Como ya les nombramos, como ya les nombrábamos, este es el seminario de cierre del proyecto FIA-PIT 2018-0045, programa de manejo integrado para minimizar los impactos de la nueva enfermedad causada por la Septoria Musiva en el patrimonio forestal de variedades de álamo en Chile. Vamos a partir de inmediato con las palabras del director del proyecto, el doctor Francisco Zamudio. Profesor, muy buenos días, gusto saludarle.
Buenos días. Ustedes me escuchan bien, supongo bien, Juan Pablo. Bien.
Sí.
Primero que todo, unas breves palabras. Debo partir excusando al representante de la Fundación para la Innovación Agraria, FIA, quien no pudo asistir. Él era la primera persona que iba a dar las palabras de bienvenida a este seminario de cierre. Por lo tanto, lo estoy realizando ahora. Primero que todo, quiero agradecer a la Fundación por su apoyo constante durante los 4 años que duró este proyecto, un año extra, debido a razones que ya voy a mencionar un poco, pero obviamente nosotros ya intuimos, ¿cierto? La pandemia nos afectó a muchos, en nuestros trabajos y en nuestras vidas, en este proyecto también. Segundo tema, quiero agradecer la asistencia y la colaboración y la amistad que he cultivado durante años ya con Marisque y con Richard. Los voy a presentar a ambos. Quiero también agradecer a la Dirección de Tecnología de Información por su apoyo técnico en que este seminario de cierre resulte. Y para dar inicio entonces, quiero por favor indicar más o menos cómo nos vamos a organizar. Primer tema es la primera presentación va a estar a cargo de Mariske Steniker. Ella es investigadora, es científico, investigadora, bióloga, forestal, genetista y patóloga y un montón de otras cosas más relacionadas con álamos en Bélgica. Ella trabaja en el Instituto de Investigación en Bosques y la Naturaleza. Pequeño comentario para conocernos un poco más o conocer de ella. Tuve el honor de conocer a su padre, don Victor Steniker, un brillante forestador, un brillante genetista, quien fue una de las personas clave en el desarrollo del álamo, de las nuevas variedades de álamos en Europa, y su hija, Mariske, ha continuado ese trabajo de él, ese legado familiar ya. Ahora, por otra parte, la segunda presentación va a estar a cargo de Richard Hamelin, un gran amigo, ya de años ya, que está presente ahora, bueno, está en British Columbia. Él trabaja en UBC, la Universidad de British Columbia. Bien, él yo diría que es el referente en el tema de este patógeno con el cual vamos a hablar en Canadá y en general en los Estados Unidos, yo diría. Él es profesor y es jefe del Departamento de Ciencias Forestales y de Conservación, Ciencia y Conservación Forestal, de la Universidad de British Columbia. Bueno, ellos son los dos, van a hacer sus presentaciones, ambos lo van a hacer en idioma inglés. Esto significa que si en algún momento alguno de ustedes tuviera alguna consulta relacionada con las presentaciones, al final de cada una de ellas, yo puedo ayudar justamente en la traducción, en caso que fuese posible o fuese necesario. La tercera presentación es la mía, la presentación de cierre. Yo lo voy a hacer en idioma español. Sin embargo, tal como les comenté a Mariske y a Richard, yo voy a transmitir, entre comillas, en idioma bilingüe un poco. Voy a compartir con ellos algunas de las principales ideas que están en las imágenes que voy a presentar, pero la presentación va a ser principalmente en idioma español. Al final, obviamente, de mi presentación, algunas breves palabras de reseña al final y de seguimientos a todos los que han colaborado con nosotros, mis co-investigadores y asesores. Juan Pablo, por favor, demos inicio ahora.
Excelente, profesor. Muchas gracias por sus palabras de bienvenida. Vamos a partir con la presentación. Bueno, mi inglés is not so good. The influence of climatic change on the spread of disease and pests in Europe and consequently of breeding, de la doctora Mariske Steenecker, Research Institute Nature of Forest, INVO, Flanders, Bélgica. Esta presentación va a ser en idioma inglés. De todas maneras, muy buenos días, doctora. Muchas gracias por su participación el día de hoy.
So good morning or good afternoon. Sorry, this is the last slide. I have to start with the first one. Okay. I would first like to thank the organizers of the seminar who extended the invitation to me to share some views and comments about popular breeding in Belgium and the influence of climate change on the spread and incidence of popular diseases and pests in Europe. And speaking about climate change, well, in July we had the hottest day ever measured in Belgium, but also in the rest of Europe, it was boiling hot, 39 degrees or Celsius or 102 Fahrenheit was recorded. hybridizations. They are fast growing, but we don't have much experience with them. But up till now, we are very, we have several clones on the market, and these hybrids again have been made with a Populus trichocarpa deltoides, and we crossed them with a Populus maximovici to increase the tolerance to rust mainly, but also to Marsonina brynea. So that's the reason why, but they are also very fast growing clones. And then the peg crossings, again, we made them because in order to increase the tolerance to rust. So we had very good hybrids, very fast growing hybrids, Trichocarpa deltoides, but because of the high susceptibility to rust, we were not able to use them, but we could use all these good characteristics by crossing again with the Populus deltoides with a good tolerance to rust. So that are the different hybrids that we are making, and that we've been making, and we're still making in our breeding program. We have at the moment, at the last 20 years, we commercialized 9 different poplar cultivars. Several of them are used all over Europe. And the most recent one was put on the market in 2021, and they're all protected by breeders' rights. So what did we do from 2000 till 2014? Again, we started to make controlled hybridizations in our greenhouse, and we made 700 controlled hybridizations, from which we obtained 95,000 seedlings. It was a huge work, and all these 95 seedlings, let them grow in our nursery first. And where we observed them for resistance to the different diseases. I will explain more about the diseases later on. We observed their phenology, the moment that they start to grow and when they stop to grow, in order to know if they are good adapted to our climate. We look for the rooting capacity of the cuttings. We observed their growth and the interaction of clone. And then afterwards, after this, all these characteristics, the best clones are going to field trials, where again we observe the diseases, but we focus mainly on interaction of the clone and the site and the branching of the clone. And finally, after 18 to 20 years, we also work together with the University of Ghent to test the wood quality for peeling and for sawwood. So it's in order to, before you can put a clone on the market, you need at least 20 years, consecutive years. So now from this breeding program from 2000 to 2014, we have now 53 clones selected based on all these characteristics. And we installed a popular experimental network of 120 field trials, but not only in Belgium, also in France and Spain, because we want to evaluate the agronomic performance of the clones in different environmental conditions. Also because a lot of our clones are not only used in Belgium, but in the rest of Europe. So this is in short about our breeding program. So what were the most important diseases we were selecting for until 2000? These are a bacterial canker caused by Xanthomonas populi, the leaf spot disease caused by Marsonina brynea, and the rust Melamsora populina. And other breeders, like in France and in Italy, also are selecting for Melamsora populina, especially in Italy, where Melamsora populina is the most important leaf species present in their plantations. Bacterial canker of poplar is a disease that is already present since many, many years in Europe. And when the breeding, the program in Belgium started in 1948, they started to work with Populus tremula and hybrids between Populus tremula and Populus tremuloides. And but after some years, we saw that most of these hybrids were very susceptible to this bacterium, and therefore they switched to the other 4 species I just presented. And we developed at our institute an artificial infection method which we put a drop of bacterial suspension where we grow 1-year-old trees, we remove a leaf, and on the leaf scale, we put a drop of bacterial suspension. And after the second year, we already can say whether a clone will be resistant or not in the future. And this is it seems to be a very reliable method, because all the clones that have been selected 13 years ago still are very resistant to the disease. So with this disease, we don't have a problem at the moment. And then we also, in the 60s, Marsonina brynia, the black leaf spot disease, was introduced from the States and Italy, and it's now present all over Europe. And it's a leaf spot disease that causes early defoliation of the trees, of the leaves, and with the consequence that the trees are not stopping growing very early in the season. They stop growing very early in the season, and it's a big influence on the growth. And some Marsonina, some very susceptible clones can even be infected on their small branches. Then the third one is the one that I've been talking about, is the Melamsora larischipopulina. And as I said, in the 90s, the Trichocarpa deltoides clones became very susceptible to rust because of the development of new pathotypes. And this is the interaction between the rust and the host, also the poplar, is based on the gene-to-gene model. And that means that for each gene controlling resistance in the host, there is a corresponding specific gene controlling avirulence in the plant. And that means that in total, we discovered 8 different resistant genes and in the plant. And in total, we have 2 to the 8 is 256 different combinations of these avirulence genes, which make that we have 268 different pathotypes in the rust population. And these pathotypes cannot be identified based on observations that we do in our nursery. So we have to do artificial infections in the lab with the different races separately. And this is a huge work, but it's working very well. But nowadays, we are no longer selecting clones for total resistance to rust. We now select for horizontal resistance. That means that the clone is not total resistant, but it has a good tolerance. The rust has no influence on the growth of the plant. And that's why the rust problem, which was a main problem in the 90s, is no longer a big problem in Europe, because we have a good method to select for selection of clones with a good tolerance. But now what is going on in Europe? We see that the ongoing climate change is inducing crucial changes in diseases of poplar in Europe. And already 20 years ago, several authors suggested that a warmer climate will bring an increase of diseases. And this fact is supported by a positive effect of the warmer climate on pathogen and vector growth rates, and a greater potential of these species compared with their hosts to respond to environmental change through dispersal, also phenotypic plasticity and adaptation. What are the big changes we see? I told you I will focus on the most important one we see in Europe. So we see changes in distributions of pests and pathogens. There are several pathogens and several pests that we see occurring in areas where they didn't occur before. I will focus on later. And then we also see change in intensity of infections of the two rust species. And what's very important, what we see is that we have the introduction of exotic pathogens by human-mediated long-distance transport. So what about rust? The Melamsora larischipopulina is the most important rust species in northwestern Europe. But in the course of the last 30 years, we've seen in our nursery that infections by this rust starts now 2 to 3 weeks earlier in the season. And susceptible clones, we see that as a result of that, we see that on susceptible clones, there are more infection cycles in 1 year, and the infection level of the clones are much more infected than before. And this is based on this change in the start of the infection. But at the same time, we see that the incidence and epidemic severity are, as we know, they are correlated with the relative humidity and leaf surface moisture. And so we, like the last 5 years, because of the high temperature and in our areas, we see a lower rate of infection due to warm and dry weather. So it's really very difficult to, more difficult now to select for resistance to rust. On one hand, we see that in some years the infections are heavier, and other years, because of the drought, infections are less heavy. And so we cannot only rely anymore on observations in our nursery. We have to combine with, again, with artificial infection. And in Europe, so as I told you, Northwestern Europe, Melamsora larischipopilina predominates, but Melamsora alipopilina is mainly encountered in southern regions, like in the north of Italy, it's the main rust species. But Melamsora alipopilina is favored by warmer summer climate and slightly by water deficit in the same month. So it's predicted by the Spredo store that under the climate scenario, coming years, Melamsora alipopilini will become more important in northern areas of Europe. And we already see that in the center of France. And so the situation in these areas will be, at the end of this century, will probably be similar to that in the present day Southwest. So that means that in the future, we will have to change our priorities in our breeding program for resistance to disease. And what we never saw in Belgium and in the north of France, we never had problems with pests, with insects. But the last 10 years, we saw outbreaks of infections in some of our young plantations. And this is a list of pests that have been published by Gennaro from Italy, where he made a list of all the pests that occurred in Italy since the 50s. And some of them became now much more important in some areas. And what we see not only in Italy, but also in Belgium and in France, some of these insects are more common. And the most important one at the moment is the woolly poplar aphid. Is becoming a big problem and more important in our breeding program than the rest was in the past. So the woolly aphid causes destructive infestations on poplar. The invaded trees are over 6 or 7 years old, and the first signs of attack are difficult to see because the first colonies of the infection, they occur in the crevices of the bark high in the upper third of the trunk. So in the area where lower branches are inserted. And when strong proliferations occur, colonies expand from the lower part of up to the lower part of the trees, completely covering it with a white, with a whitish felt-like wax that is secreted by the insect. And in the 90s, heavy infections generally occurred sporadically in the poplar plantations in Italy, and they had been recorded every 5 or 10 years. But now in Italy, because of the favorable climate trends, they occur more frequently. And what we saw, so until the 1990, the disease was only present, the insect was only present in Italy, but in 1996, the first heavy infections were seen in the south of France. And year after year, the insect moved to the north of France, like in 2011, heavy infections were seen in the north of France. And last year, for the first time, I saw trees that were killed by the woolly aphid in our country, in Belgium, on this trees of a very susceptible clone. So that means that all our clones that have been selecting during the years have now to be screened for resistance to the woolly aphid again. And that in the near future, we have to take, if we make new crosses, we have to know about the susceptibility of the parents that we will use in our breeding program. Luckily, our colleagues in Italy and France have been developing an artificial infection method. It's a simple infection method where they take a branch of infected trees in their nursery, and they put it together with the clone that you want to test under certain conditions of humidity and temperature. And then after 1 month, you can see whether a clone is resistant or not. But the method is not totally reliable. We see that sometimes when they do a test, they say that the clone is very susceptible, whereas in the nature we don't see any infections, and the opposite. So we still have to work on the method, and we also have to see whether there are different pathotypes present in nature. We know, for instance, that Populus nigra is very susceptible. But in Italy, they found out that Populus nigra from the north are more susceptible than Populus nigra coming from the south of Italy. And also Populus deltoides is susceptible, but Populus tremuloides and Populus maximoixi. So it will be a big job for us to start to test again our material and to select all the parents for the breeding program. And two other insects that are worrying me are the goat moth and the hornet moth, and because these are wood borders. And so the hornet moth, the one that you see here, the hornet moth damaged the root system of the poplar. So the moth drops a large number of eggs from a branch to the ground. And the caterpillars that you see here enter the tree at the base of the trunk through bark gaps or other weak spots in the bark. And then they eat their way down between the bark and the wood to about 20 centimeters below the ground level. And there they hibernate, and after which the feeding continues until autumn. And after hibernation, the caterpillar pupates into a butterfly, and around approximately 5 millimeter large exit holes are produced here, and above 50 centimeters above the ground. And the problem is when there is a heavy wind that the trees are broken at this level. And so trees of 8, 9, I've seen a plantation recently of trees of 8, 9 years with a heavy wind and the storm we had in spring, that 50% of the trees fell down and were killed because of this insect. And the same problem we have with another wood border, that is the goat moth, which the larvae make big holes at the bottom of the tree. And again, with heavy wind, the trees are broken because of the galleries that the pupates are making in the woods at the bottom. We don't know yet if we don't know anything about susceptibility of the different poplar species or the hybrids. So that's something that we have to work on. But I'm really afraid of this more and more insects coming from south of Europe to our breeding program. Another thing what I want to mention is that Xanthomonas populi was previously only present in the northwestern Europe, mainly in France, UK, and in Belgium. But nowadays we see that the Swedish colleagues are starting to plant more poplars in the south of Sweden because the climate is becoming more favorable for planting poplars. But now they are afraid that bacterial canker will move to the north to the south of Sweden, and that's why our institute is now testing resistance to bacterial canker of the clones that are used in Sweden as a preventative measure. So there are two exotic pathogens introduced from Asia and Europe recently. And one is the Brandneria populi, subspecies populi, is a bacterium causing bark canker. It's introduced from China. And then is there the brown marmorated stingbug introduced from Asia. So first about the brown marmorated stingbug, it's an insect that is native to China, Japan, Korea, and other Asian regions. And as you can see, the first symptoms, when there is a crack in the bark, you see a white exudate coming from the top from the tree to the bottom. And then it's forming afterwards a brown, stinky mess on the tree. And the bark is broken here. As you can see, you have a big damage to the tree, and on very heavily infected trees, you see that the trees will be broken or will be killed. And the first symptoms of these infections were seen in Hungary in 2012, and later on in Spain in 2016, Iran, and Serbia. And nowadays, mainly in Hungary, but also in China, we see that many thousands of trees are killed because of this insect. And mainly Populus deltoides times Populus nigra are very susceptible to this insect, to this bacterium, sorry. And then there is the Heliomorpha halis, the Asian stingbug. This is a polyphagous plant feeder. It's causing damage to a lot of agricultural crops, and it's introduced in several European countries since 2012. And in 2017, there was a serious damage to the crops in northern Italy, to agricultural crops, but a lot of poplar trees were also attacked in the nursery of the breeding program, but also in the plantations. So introduction of exotic pathogens has been a problem since I started to work at my institute, but it's becoming more and more important in our and a big problem for our breeding programs. So to conclude, I want to say that when you create a new plantation, you should choose your cultivar not only based on vigor and shape and wood quality. Of course, this is very important, but especially take into account the resistance tolerance of the cultivar to disease and pest. And at the same time, when you install a plantation, plant different cultivars, not only one as we did in the past in Europe in the 90s, where 90% of the plantations were made with just one cultivar that became susceptible to rust. So it gives you more certainty if you plant different cultivars, if new pathogens appear in the plantations. And to nurseries, I want to tell that use best management practices to reduce the risk of introducing unknown pests. Luckily, we know that in our breeding program, we can use, there is a suitability of a genetic control of resistance and tolerance. And nowadays, breeding programs undertake research for quantitative resistance, recurring to molecular markers. And we now can use also associated with studies on epigenetics and also microbial communities of various tissues through the new approach offered by metagenomics. So I'm convinced that we can, with breeding, we can continue to bring good clones on the market, but it could help if not too many new diseases will be introduced in Europe. So thank you very much for your attention.
Thank you, Mariske. Thank you very much. No sé si habrá alguna consulta que quieran hacer a Mariske. No he visto el chat. Ya, pareciera que no. Entonces, Juan Pablo, procedemos a la siguiente presentación. Bien.
Bueno, agradecerle a la investigadora por la interesante presentación, excelente. De todas maneras, si hay algunas preguntas, al final del seminario se pueden realizar sin problemas, también se pueden escribir por la vía chat, si es que les interesa, y por supuesto que también dejaremos el correo del proyecto para las preguntas también que estime necesaria posterior. Así que vamos con la segunda presentación.
Sí.
Profesor, cuando usted diga, si quiere hacemos la presentación, usted quiere decir
Sí.
algo, pero partimos cuando usted diga.
No, justamente veo la cámara que Richard está fuera de la cámara pero esperemos un minuto para que él pueda retornar y dar inicio a la presentación que nos tiene preparadas. ¿Ya?
Bueno, por mientras vamos a ver el nombre. Esta es la segunda presentación de esta jornada. Tiene como nombre Novel Approach to Improve Our Understanding of the Septoria Care of Poplar, de Richard Hamelin. Él es PhD, profesor en Head de la Facultad Forestal, Department of Forest and Conservation Science de la Universidad British de Columbia, en Canadá. También será esta presentación en idioma inglés. Richard,
Sí.
buen día.
Thank you, Richard. You can go ahead. Thank you.
Okay, can I share? Let's see here. I need to share my, oops. Okay, share screen. All right, can you see my screen now? Perfect, perfect. Well, thank you very much for the invitation. It's a real pleasure to talk to you today. I would love to have gone back to Chile. I was there a few years ago, pre-COVID. I visited with Mauricio and Francisco. That was a fantastic trip. Made great friends and also developed some interesting new research. And so I'd like to share some of these the research that we've been doing in the past few years. Picture that you're seeing is, well, I'm from Quebec originally. Now I live in British Columbia in the city of Vancouver. I'm a professor at the University of British Columbia. And this is sort of in my backyard. So we have the mountains. Vancouver is a beautiful city. You have the ocean on one side, you have the mountains on the other side. You can literally drive 30 minutes and be up in the mountains and see these beautiful, beautiful landscapes. So it's a great place to be. And also I wanted to tell you this because a lot of the what I'm going to be referring to contrasts plantation forestry with natural forests. And this is, I think, a big issue. And in Canada and British Columbia, we have a lot of natural forests, but we also have planted forests. And I want to contrast the disease problems that we encounter in these completely different environments. All right. Maraki already mentioned some of this, but we are living in the age of the Anthropocene. That's where the planet is changing in ways that are irreversible, or we think are irreversible. Maraki already mentioned climate change that increases tree stress, promotes some pathogens, as we heard. She also mentioned invasive species, things that we are moving around that take advantage of new empty niches, and also hosts without resistance, which is very relevant to our poplar story today. And also this is something that particularly in Canada we deal with, forestry practices, logging and planting. This is a very human-driven practice. The way we cut, the way we replant, really affects the forest health in the future. Let me see if I can move this thing around here. All right, okay, all right. Sorry, I'm just trying to reorganize the icons so that I can see my slide. Okay, so these kind of plantations that you have in Chile and in many places in the Southern Hemisphere. That's where in British Columbia, where I am right now, mostly we don't have that. We have natural forests, multiple species of trees growing, but there are some plantations, more and more to produce one particular type of tree for specific purposes. That's where, some of the disease problems arise. Now, forced health monitoring is crucial to stay one step ahead of the problem. And, we do a lot of that in Canada. So doing surveys and inventories to make sure, like Marijke mentioned, that we can detect these potential, pest and disease outbreaks before they get established. Because once you have a big problem everywhere, it's really hard to control. And, so establishing permanent plots, having forced health experts to do surveys so that they can provide advice on early intervention, and also for future record, because Marijke mentioned some of these pests were already present in the 1930s, so people knew about them, but then they flare up now, possibly driven by climate change. So these are the kind of records that we need to get better at providing. So of course, some of these invasive species, they can, decimate entire host population. As I said, once they get introduced, they can spread and they can affect entire ecosystems. So we have very, very famous, infamous example, the Dutch elm disease, the white pine blister rust, chestnut blight, sudden oak death. These are all pathogens that have in common that they were exotic. They came from elsewhere to North America and elsewhere as well. And they established in forest ecosystems, and they completely modified the forest ecosystem. And I like this picture here. This is a grizzly bear, of course, that we have in Canada. And the grizzly bear, actually, probably a lot of you don't know that, but grizzly bear are threatened by this very disease here, white pine blister rust. And that's because in high elevation, the only tree species that can grow are white bark pine, and white bark pine is threatened by white pine blister rust. As it turns out, the bear feed on the pine nuts. The pine nuts are full of calories, and the bear gorge on pine nuts, late in the season, so that they can go hibernate. And because of white pine blister rust, these pine nuts have become rarer and rarer, and the grizzly bear then go down in the valley, and that's where you get encounters with other animals like humans, for example, and that never ends well for grizzly bear. So, there's a direct link here between an exotic disease and the survival of a species like the grizzly bear. Okay, so moving on, I want to talk to you about some of these challenges that we're facing, especially in terms of being able to recognize, pathogens, being able to identify them at different life stages. Sometimes, you'll have just, symptoms on the foliage. Sometimes you'll have, a little discoloration of the bark. And, to be able to act rapidly, we need to be able to have a quick diagnostic. And that's very challenging. This is my friend and colleague, Nicolas Faux, who visited Chile with me a few years ago, and he's an expert on these pathogens. He did his PhD actually on poplar canker. The other thing that's important, Marijke mentioned shifts in pathogen, races that can overcome resistance. So that's really important, being able to detect those shifts. Also tracking sources, because a lot of these pathogens move around, and being able to track the source methods, and also selecting resistant hosts, because often where these pathogens come from, that's where you find resistance in the hosts. Okay, so I'm going to tell you about genomics, because there's been a revolution in biology really in the last few decades, and that's genomics, our ability to decode DNA in living organisms. And that really has changed the way we do biology in general. So we can now do things like, look at, decode the genomes, look at gene expression. We can modify genome, edit genome. We can do fantastic things. And we just at the beginning of this genomic revolution, but we have all seen during COVID how central genomics became to our answer to the COVID pandemic, from the mRNA vaccine to the PCR testing to the genomic surveillance to identify variants. These are all things that genomics allowed us to do to fight this terrible pandemic. And my point is that we can use the same tools in trees. And I want to tell you a little bit about this, these genomics application in forestry. So similar to COVID, we also have been doing pathogen detection using DNA-based PCR. We've been doing that actually before COVID. So, most people have never heard of PCR before COVID, but we've been working on PCR detection of pathogens since the 1990s, really. That has really helped us to in our ability to diagnose plant pathogens. DNA barcoding and metabarcoding, these are kind of spin-offs of PCR. Basically, you amplify a piece of DNA, then you decode it. That allows you to identify unknowns or at least match the unknowns with with databases so that you can put names on new pathogen. And this genomic biosurveillance is very powerful. We've seen with COVID how the variants, shifted over time, and it's through this genomic surveillance, whole genome sequencing of the virus, that they were able to do that. And this is something we're doing with pathogens, and I will tell you a little bit more about that. So that's what genomics can do for us: discover and monitor variants, identify sources and pathways, predict traits also. So this is very important, and we've done some of that with poplar, especially with regard to resistance in the host. So poplar resistance to rust and poplar canker, finding genetic markers that are associated with the resistance. So this is very, very important. And then the tools for biosurveillance. Okay, let me tell you a little bit about poplars. We got a great introduction from Mareike. But look at this map here, North America. Poplar is an amazing tree. It grows pretty much everywhere in North America. This is not planted. This is natural occurrence of poplar in North America. It goes from coast to coast, coast to coast to coast to coast. These are, trees that can adapt to just about anything. We find them in the boreal forest here, very much at the edge of where trees can grow. We find them all the way in the desert, in Arizona. I've seen some of those poplars in the Arizona, Populus vermonti. You can see that in Canada, like every province has a poplar, a lot of Populus balsamifera, a lot of Populus tremuloides. And then on the West Coast, then we have Populus trichocarpa that has been so popular in the breeding program. So that the clones, the hybrids that Mareike was talking about are between deltoides. Let's see here, where's deltoides? Yeah, the yellow one, deltoides, and trichocarpa. I'm not sure, maybe I should ask Mareike about this, but why did they not use balsamifera in some of those crosses? Maybe she can tell us later. Poplar has a lot of pathogens. Actually, there's a book written on poplar diseases. There's about 200 different diseases of poplar, and We've heard about some, but the one that I wanted to focus on today, because it's what brought me to Chile, and it's brought me to collaborate with Francisco and Mauricio, are the Septoria pathogens. And I apologize because pathogens tend to change their Latin names all the time. And what we used to call Septoria Musiva is now called Spirulina Musiva. And you'll see in my presentation that I'm also calling it Mycosphaerella populorum, because for a while that's what it was called. So it's all the same thing. We call it Musiva and Populicola. And so there's two pathogens that are native to North America that attack poplar: Spirulina musiva and Spirulina populicola. And in the natural environment, what they do is they cause a leaf spot. They don't cause cankers. They only cause leaf spots in a natural environment. So they rarely kill trees. So part of the natural ecosystem, the defoliation happens anyway. They're deciduous trees, so in the fall the leaves fall. So in a natural ecosystem, it does not threaten the survival of the trees. So not a big problem. But in related to our domestication, cankers have started causing these deadly, the breaking of the trees, of the stems, and the attack of the cambium tissues. And you can see this is a picture I took in Chile. Actually, there's these cankers are extremely damaging. They also affect the wood quality, the wood color, and then eventually, branch mortality, tree mortality, and then some breakages. So very, very important disease of poplar wherever it's present and wherever susceptible poplars are grown in plantation. And this has very much to do with this poplar domestication. The disease, as I said, was present in North America. No one worried about it until the beginning of the 1900s, maybe late 1800s, beginning of 1900s, when actually farmers in the Midwest of North America started growing poplar as windbreakers. And that's when this disease started showing up. And then eventually, poplar was grown in clonal plantation, and then the disease really exploded. And one of the challenges is that there's a large reservoir of the pathogen in the natural stand, because as you saw on the map, these Populus deltoides have a large distribution. They occur naturally. And so there's a large reservoir of the pathogen itself, near where you plant poplars in plantation, like in the Midwest, in Kentucky and Tennessee and Missouri and these places. So the pathways of introduction, it's pretty clear that it's through propagation of clones cuttings being shipped around. There's some good evidence that the pathogen can survive in either buds or inside of the infected tissues, maybe below the bark and the cambium. And then once the trees start growing, the pathogen is there and then can start infecting the leaves, and then eventually cause cankers. So almost certainly the spread has not been natural. It's been aided by our spreading of trees through vegetative cuttings. So we did a project. So prior to my visiting Chile, we had worked on this question of where the pathogen had originated from, because Septoria musiva is believed to be native here in northeastern North America, overlapping the range of Populus deltoides. So it was not present anywhere else. But like I said, about 100 years ago, shows up in Saskatchewan. Maybe 30, 40 years ago, shows up in Alberta. And then maybe 15, 20 years ago, it shows up in British Columbia. So we try to understand if we could identify the source of this of the pathogen through this genomic surveillance approach. Now, there's a couple of suspected sources. First of all, in the Northeast North America, Quebec and Ontario have had big popular breeding programs, doing selection, doing crosses, and then sharing material. The other big place is Minnesota, Wisconsin. The Americans also have had big efforts since I guess maybe the 1960s, 50s and 60s, all the way to the 70s, 80s, again, breeding poplar, producing genetic material with the desired characteristic, and then shipping it around. And then the last is Saskatchewan. They also have developed a breeding program where they have produced genetic material that they have shipped. So these are some of the potential sources we were thinking about in our study. And since I went to Chile, then we decided, well, let's include the Chilean population there, so we can maybe pinpoint where it originated from. So the first thing we did is we worked with the Americans, the Joint Genome Institute, to sequence the poplar canker genome, and we did that, and we published this in the journal PNAS in 2015. And I don't want to bore you with the details of the genomic analysis. This is something I can get excited about, but I'm not going to say too much, except that we have the genome of this pathogen is very sort of simple in a way, and well organized. We have 13 chromosomes, and the chromosomes are sequenced almost to perfection. The little red dots here show telomeres. So we have, for almost all of these scaffolds, we have the entire sequence of the chromosomes. So from telomere to telomere, so really, really high quality genome. And then already you can see that we compared the Musiva and Papilicula genome. You can see that there's some differences. We wanted to focus on those differences because we want to understand why Septoria musiva became a canker pathogen of poplar and not Papilicula. And so this is an important point. So we did some phylogeny. So as it turns out, relatives of Septoria or Spirulina can attack a bunch of different plants, from tomatoes to bananas to pines to wheat. There's some microspirilla type of pathogens that affect all of these hosts, and we wanted to see, to look at the host tracking. And as it turns out, sure enough, Papilorum, the two popular pathogen, Musiva and Papilicula, are much more closely related. But you can see that the other pathogens of pine, tomato, and banana are also phylogenetically or evolutionarily related to the poplar canker pathogen. But what's really cool is that we can actually, using this genomic data, we can estimate the time of divergence to a common ancestor. So we can basically go back to the root of the ancestor of Papilicula and Musiva, and we can estimate how long ago that happened. Turns out about 6 million years ago. So Papilicula and Musiva were together with a common ancestor about 6 million years ago. And as it turns out, this is the same range of time where the Papilus deltoides also diverged from Papilus balsamifera in North America, occupying completely different sides of the continent. Balsamifera going north, deltoides staying northeast. And so that split, it looks from this analysis like the pathogen basically kind of specialized one on balsamifera and one on deltoides. And the other thing that last little genomics slide here from that PNAS paper, we wanted to understand why Musiva can attack the woody tissues, because that's what makes it such a bad pathogen. If it was just foliar, well, it's bad also because the rusts are a pathogen that can cause heavy defoliation. But once you get a pathogen that attacks the stem, then you can have major damage, especially when you have so many cankers as in the picture I showed you. It can be devastating for a plantation. So we wanted to understand better why the Musiva is able to attack the woody tissues of the poplar, but not Papulicula. Both of them can attack the leaves and cause these leaf spots, but only Musiva has learned or adapted how to attack woody tissues and not Papulicula. So as it turns out, when we compare their genomes, we have an expansion. So the genes that are involved in cell wall breakdown, so things like cellulases, hemicellulases, are increased or expanded in Septoria or Spirulina musiva compared to Papulicula. And also the genes involved in cell wall breakdown are expressed much more in Musiva than in Papulicula. So you can see here, this is gene expression of those genes, and then Papulicula much lower and even decreased gene expression of those cell wall breakdown enzymes. So that gives us sort of a clue as to why Musiva has evolved to attack woody tissues. So these, proliferation of genes that can break down cell walls in the woody tissues, because we know that the fungus actually penetrates inside of the xylem woody cell. So we have really cool microscopy that shows the fungus is actually inside the woody cell. So it's not superficial, it's deep in the wood. Okay, I want to tell you something about, because I want to tell you about the Chilean samples that Maurizio and Francisco sent to us, and I want to tell you about this global population genomics of Septoria, Spirulina musiva. And so what we did is we sequenced hundreds of genomes of the pathogen. So we got the pathogen in pure culture, petri dishes, from everywhere in Canada, from the US, and then we plugged in these Chilean samples to see how they would compare. And these are a representation, like either through a phylogenetic tree or with a haplotype network. And you can see that there's different, and I colored the samples. Each little dot here is a sample, like a culture of the fungus. And you can see, I color them with their geographic origin. And you can see the colors really stick together. So that's good news, or it tells you that there's some geographic pattern or geographic fingerprint or genomic signature in the pathogen. And what's really cool also is that you can basically kind of follow geographically the evolution of the pathogen. For example, here you see that's British Columbia, that introduction from 2005. Here you can see we have Alberta. We have another, this is British Columbia from the Okanagan, the second introduction. And here we have Alberta, and then Saskatchewan. So if you follow the map, you're kind of going from BC to Alberta to Saskatchewan. So kind of this stepwise mutation model applies. And here I put USA all together just for this map here. You can see the USA population is kind of the backbone of this network, and that means it's likely that most other samples derive from this backbone. And then here you can see Eastern Canada, and here you can see Brazil, and then Chile all the way down here. So Chile, very different from, well, they have their own genomic signature, and that's to be expected if it was introduced. But it looks like Brazil, unfortunately, we've had just one sample from Brazil. We have no sample from Argentina. I know that the Chileans are very interested in knowing if they if they got it from Argentina. I think if we did have Argentinian samples, we could answer that question. But it looks like there's a step between the American and the Chilean, and that step, certainly Brazil is in the step in between. So it'd be really interesting to see where Argentina fits here. And just sort of to highlight the point that I'm making here. So I also want to say that the probable source of the Chilean introduction, well, if we can exclude Brazil and Argentina, they may have gotten it first, but where did it come from? So I think we can exclude Saskatchewan, because we know that Saskatchewan, they have sent cuttings of poplar to a lot of places. I think we can exclude it because it's so far in this phylogeny. And Canada, Eastern Canada, Quebec, and Ontario, they're not that far, but there's many steps in between. So I kind of think we can exclude them. Canada, BC, certainly completely different. But it's the Midwest, really, the American Midwest. And you can see that this kind of backbone that leads into the Chilean population. And of course, this here is Brazil. So to me, the most parsimonious explanation for these results is that Chile got Spirulina musiva from the American Midwest via Brazil, possibly Argentina. At least that's what this result tells me. Okay, and this is, sorry, the last thing, the USA East is actually quite different. So both Quebec, Ontario, and USA East South, that's like I'm talking here about Kentucky, places like that, Kentucky, Missouri. So it really looks like the Midwest is the connector here. All right, so I want to just finish by telling you a little bit about something we've been developing and using for the poplar canker, and that is, and I've been always passionate about this, taking genomics to the field. And because especially in Canada, well, I'm sure most places, people are really interested in being able to do the DNA testing themselves on site. And that's because a lot of time you will be driving to plantations or to reforestation sites, and you want to be able to do your testing to have quick results in the field, instead of having to bring samples to a lab or ship it to a lab and then wait for a week or two or even more to get your results. So we've developed a method that basically you can do your DNA testing in the field. All the material and the equipment needed, it fits in a backpack. And I did a little demo here with the news media organization, CTV News, and took them to the woods. And this is a Douglas fir stump, and that's my lab bench. And everything that you see here that's the only thing I need to do a PCR test. And so you've got an instrument. I'm going to go through, show you all the what is needed, but you can really do your DNA testing in the field, and we've applied it to poplar canker because the BC province, the provincial Department of Natural Resources, they want to know if the poplar canker pathogen will spread to natural Populus balsamifera and Populus trichocarpa. That's their biggest worry, that it will spread into the natural stands and then potentially affect the riparian species survival. So they want to do some testing, and so we developed this method for them to use, and they've used it in hundreds of samples going in the fields and testing around plantations in British Columbia where the popular canker is present, and testing the natural stands of the black cottonwood Populus trichocarpa in the surrounding to see if the pathogen has spread. And so this is just going through the material. Oops, sorry. It's basically a PCR reaction, same as you do with the COVID. We have a portable instrument that's battery operated. You could just charge it, take it to the field. Battery lasts long enough for a day of work. And what our innovation is to develop these lyophilized, ready-to-use reagents that are stable at room temperature. So you can keep them in your backpack for up to a year, and they will not go back. So you don't need a refrigerator, you don't need centrifuge. Everything is lyophilized, ready to use. All you need to do is add your sample. And we have a very simple extraction buffer, and it turns out to work really well for samples like, well, we've worked on insects. So this is the gypsy moth, now renamed spongy moth. And like I said, we've used that with poplar leaves spots to identify or to detect Musiva and separate it from Populicula, because the leaf spots are identical in the two. So you cannot tell apart the two pathogens just from the leaf spots. So this is super important for the BC Ministry of Natural Resources, because they want to know if the leaf spots that they're seeing in their balsam and black cottonwood are actually Musiva or Populicula. So that's what they've been using this for. And so you basically take your little sample, little piece of leaf, put it in your tube, and then put your samples in this portable instrument, and then you can directly amplify your DNA by PCR. And since it's a PCR that uses a fluorophore, you can just read your results by quantifying the fluorescence in your tube. And in this case here, you can see the positive shown by this curve here. In the sample, and you can, so because you're using your iPhone for monitoring the reaction, so you can basically collect your samples, put them in the thermal cycler, and then you can walk away and then do some more sampling, and you can monitor your reaction from your phone. So, and as I said, we've developed that for multiple pathogens, from the root decay fungi to needle blight to the poplar canker, and that's already published and available. So I'd like to end on this note. Gracias. It was a pleasure to talk to you. This is another view from the mountain up here where I was hiking last week, and I will be happy to host anyone who wants to come and visit our beautiful country and city.
Thank you, Richard. Thank you for your excellent presentation. Muy agradecido de vuestra presentación. Profesor Zamudio, no sé si usted quisiera hacerle alguna pregunta sobre la presentación del profesor Richard para poder seguir con esta jornada.
Sí, solo una consulta, Richard. Just actually a question. People from Argentina phone in that they didn't have a chance to contact you last year to see the chances to send you some.
No, I never heard from them. I contacted the people that I had received the information for, and never heard back from them. So if there's a chance to reconnect or to connect with someone new there, I think that would be really interesting.
Yeah, because what I will talk after here right now today, Christian, do you remember Christian Espinosa? He was unable to attend today, but he is very much in contact with the Argentinian colleague there. He had a very close relationship with several researchers. So I will put him in contact to you to see the possibility, because if your project is still going on, there is a good chance that they can provide you with some material. That's all.
That would be interesting, yeah, because it would complete the story. Because now it looks like it's the Midwest is the original source, but there's a missing link.
Yeah.
That because of the Brazilian samples, it looked like there's something between Chile and the Midwest.
Yes.
And I think from talking to you all, it sounds like that's what you believe happened.
Yes.
So I think it'd be great to fill in that gap, the missing link.
Right. Great. Thank you.
Excelente. Thank you very much, Richard. Ahora vamos a pasar a nuestra tercera presentación. Profesor, por favor.
Mauricio tenía una consulta, creo yo.
Sí, es solamente una duda, nomás nada más, cortito. Hi, Richard. So nice to see you.
Nice to see you too.
And thank you for your presentation. It was very clear, and we know a little more about our Chilean isolates. But I want to ask you about did you have the chance to test the virulence of the Chilean isolates in your popular?
But that's it's something we want to do. We have not been able to do it, in part because of COVID. So we did all of this during COVID, and we did not have access to the labs. But I think that would be really important to do, because as you know, when we visited you, we were really surprised. These are the worst cases of septoria canker that we have ever seen. I mean, it's worse than what we have in Canada. So the number of cankers on a tree, we just could not believe it. So it would be really interesting to do that. Yeah, thanks for bringing that up, Mauricio. We talked about doing it, then COVID happened, and then we did the genome sequencing. That was easy to do, but the inoculations, it's more complicated to do inoculations.
I know.
Yeah, you need to grow the cuttings and the greenhouse, and then it's just a little more involved. Yeah.
Okay, thank you.
Thank you.
Excelente, muchas gracias. Muchas gracias, Richard. Profesor, antes de su presentación, por supuesto que la vamos a nombrar. El nombre de esta próxima y última presentación es Principales resultados sobre la selección genética y conclusiones del profesor Francisco Zamudio de la Facultad de Ciencias Sociales de la Salud, Universidad de Talca, en idioma en español. Profesor, adelante.
Gracias. Voy a ver si me funciona todo. Espero que sí. Ok. Asumo que ustedes pueden ver mi presentación, ¿cierto? Ok. Bien. Pantalla completa. Fine. Como comenté al inicio, mi presentación será entregada principalmente en idioma español. Sin embargo, hay algunos comentarios en inglés para orientar un poco a mis colegas, para seguir un poco la idea o enfatizar algunos puntos. ¿Ok? Bueno, este es el nombre es el título, el nombre de nuestro proyecto, un proyecto financiado por la Fundación de Innovación para la Agricultura, para la Innovación Agraria, perdón, del Ministerio de Agricultura. Este era un proyecto originalmente que duraba hasta el año pasado, hasta el 2021, pero fue extendido debido a los problemas de la pandemia. Ustedes ven acá abajo, el título también está en inglés. Un programa de manejo integrado. Ya voy a mencionar un poco, hablar esto de integrado. El título de mi presentación son principales resultados, como seminario de cierre. Principales resultados, pero con un énfasis en la selección genética y en las conclusiones generales del proyecto. The main reason for my presentation is just to give you the main results, okay? But with some emphasis in the genetic selection and the general conclusions of the project. Veamos un poco de información preliminar, basic background, a pesar que ya hemos visto algunas cosas que son más esenciales de lo que ha descrito Richard. You already, Richard, gave us a little bit more information about the pathogen, the genetic structure or the phylogeny of the pathogen. Vamos al año 2014, el patógeno fue encontrado en Chile a través de una típica prospección forestal periódica que hace el Servicio Agrícola y Forestal. Este fue el primer reporte en 2014. The pathogen was found in Chile in 2014 by the typical field inspection conducted by the Agricultural Service. These guys really have a good eye, believe me, they have a very good eye. And they communicate this information to the General Agricultural Service. Empezó obviamente un seguimiento del patógeno. Bueno, la sintomatología ustedes lo saben mejor que yo, ¿cierto? El patógeno ataca el fuste y también se presenta en las hojas, y en particular en el fuste en nuestro país, a diferencia de su lugar de origen, como dijo Richard. Well, you know the symptomology better than I, and in Chile you already observed, Richard, marisque in Chile is, you will see some pictures, the attack in the stems is brutal, yeah, very much aggressive. Okay, esta es una imagen general de el área que aún está siendo cubierta por el patógeno. Cubre nuestras dos regiones, de Maule y región del Biobío. This is a general view of the political distribution, you know, the two main regions in the country where the pathogen is, according to my latest information, still concentrating in this area. Both of you here, we have a Talca is around here, and the area that I'm going to describe my research is around here in the close to this border between the Maule region and the Biobío region. Both of you visit this area years ago, so maybe you can remember more or less the Central Valley, etc., north-south. I would say it's close to 500 kilometers north-south. Yeah. The area, the span area of the pathogen. Más o menos cubre una longitud de unos 500 kilómetros norte-sur, donde aún estaría concentrado el patógeno. En el año 2015 nosotros encontramos el patógeno en los predios de la empresa agrícola y forestal El Álamo, nuestro principal colaborador estratégico de nuestros proyectos, y en particular de este. In 2015, the pathogen was found in land, in forest, managed by the company that managed the largest surface of poplars in Chile. Both of you as well, we know, Richard Marije, you visited this area years ago. Is the area of the country with the largest industrial lands covered by poplar plantings. Yeah. La superficie total del patrimonio de la empresa, esta información está sacada en su página web, 3.000 hectáreas, de las cuales 2.600 hectáreas están cubiertas por bosques manejados de álamo, de distintas variedades. A total of the 3,000 hectares, the surface of the total land owned by the company. Out of this, 2,600 hectares are covered by poplar plantings in this area. Bueno, acá hay una foto. Estas fotos fueron tomadas el año 2015, cuando se empezó a detectar la presencia del patógeno. Como digo, ese año fue detectado, y nosotros vemos que el ataque a través del cancro es extremadamente severo, muy agresivo. Anilla el fuste y obviamente te lo debilita profundamente. And this is some few pictures from 2015, and we see the outrageously severe damage by the canker, and the girdling of it's obvious, right? start girdling the stem and debilitating, the structure of the tree, and very easily, with a little bit of wind, and that's it. It fell down. So, en 2016, justamente, aquí hay esta es la fotografía que mostró Richard, esta es una imagen que él obtuvo. Nosotros visitamos esas plantaciones jóvenes, estamos hablando de bosques de 2, 3, 4 años, no más allá, y empezamos con la ayuda de la empresa, del personal de la empresa Café El Álamo. Ellos empezaron a observar los daños, empezamos a interactuar con ellos. Cristian está acá observando el comienzo de los daños en estas plantaciones jóvenes. Richard, you are here right now, so we can remember your visit. We started putting our eyes in the stems and also in the leaves. Pero al mismo tiempo, ese año se presentó el ataque, la introducción al país de una plaga muy complicada, produciendo un daño muy severo, un defoliador, es un díptero, un Leucoptera cinuela, presente en Europa, nativo de Europa, Italia, me parece, de forma muy agresiva. Es un defoliador, un succionador de tejido vegetal, y que presentaba un ataque extremadamente severo. Y el otro también se presentó un ataque, ya estaba presente un ataque de una polilla nativa de Chile, cuya presencia también aprovechaba el ataque, la presencia de cancro, y penetraba con sus larvas, proliferando dentro de eso. Entonces al final el daño se mezclaba un poco. In 2016, we detected, the Agriculture Service detected the presence in the country of this dipter, tiny dipter. However, it's extremely aggressive. Obviously, natural selection, no predators, so this tiny dipter was spreading around the central valleys, north-south, from Santiago to the south. So it's extremely aggressive attack, defoliation, eager to, was sucking or whatever it could find in the leaves. But it started affecting the trees at the same time that we are starting observing the attack of the Septoria canker. But at the same time, here we have a confounding effect of a native moth that adapted very well to the presence of the canker, I would say, and started penetrating through the canker and continue attacking the tree. So sometimes we have a mixed confounding effect of this Septoria, or the effect of the larvae of the moth eating the inside of the tree. Esto es una imagen 2019, un estudio muy rápido que se hizo a decenas de árboles que fueron volteados para ver los daños internos y potencialmente el daño económico. Ustedes observan que aquí hay algo que lo vimos claramente, lo he dicho claramente en mis análisis. El patógeno entra, ataca y queda en suspensión su ataque. Después el tejido sigue creciendo del individuo y este daño queda inmerso dentro del fuste oculto, y que después se puede reflejar cuando sacamos las tulipas de madera, ¿cierto? Para poder producir, para poder ver los daños internos. This image is 2019, of course, and a survey of some trees that the company failed to analyze the potential economic damage produced by the internal attack of the tree, of the pathogen. The pathogen attack 1 year, 2 years, and then the trees continue growing and covering the damage. Apparently there is no damage outside, but the economic damage is inside. En el año 2014 nosotros establecimos un ensayo muy grande de clones, era parte de nuestro trabajo de investigación con la compañía, y esto fue producido justamente un par de años antes, un año antes que fuera encontrado el patógeno en el país. Y esto nos dio obviamente la oportunidad de poder estudiar el patógeno in situ, sin necesidad de establecer un ensayo para poder evaluar la patogenicidad del individuo. In 2014, we established our very large clonal trial. I will describe it right now. Just 1 year before the pathogen was found in Chile, so we immediately realized that we have a lab, natural lab, to start observing the pathogenicity of Septoria. Esta es una imagen general visual aérea de las dimensiones del ensayo, un ensayo de 4 hectáreas muy grande. Yo creo, dada toda mi experiencia trabajando en selección genética, yo diría que este es uno de los ensayos más largos que he visto en mi vida, clonal de álamos, que incluía 124 clones. Aquí hay una descripción, 24 clones europeos, 7 habitualmente utilizados en Chile, 93 variedades que yo traje de los Estados Unidos, y principalmente uno de borde nada más, pero es irrelevante mencionar ahora. Yes, this is a general view from the air, from a plane, just describing the dimension of the trial. I would say that, given my experience, this is one of the largest that I've seen in all my years visiting different countries, observing the popular programs around the world. 4 hectares and 24 hybrid, different origin, 24 generating from Europe, 93 from the United States. I will describe the pedigree a little bit. And 7 clones that were already established in Chile. We use them as a kind of genetic control for comparison purposes. 20 blocks, 20 bloques, 124 variedades. Ok, esta imagen, esta tabla nos muestra entonces los 10 tipos distintos de pedigree. We tested 10 different types of pedigrees, en total de 124 variedades. Pero mi análisis genético estadístico se concentró solamente en 4 variedades, 4 tipos de variedades debido a los números. Para análisis genético estadístico preciso es conveniente mantener un mínimo de valores. Eso ya lo he aprendido con mucha experiencia y dolor en ese análisis. Marisque ya nos ilustró un poco el significado que tiene estas letras. La primera letra es el taxón de la madre y después de la X el taxón del padre. Deltoides nigra, y acá tenemos un retrocruzamiento, como mencionaba ella, TD cruz D, TD cruz T. Tenemos variedades en términos genéticos, hablamos de F1, generación filial 1, tricocarpa deltoides. Como digo, me concentré en análisis de estos 4 tipos de pedigrí. So just a very general description. I established this trial with 10 different types of pedigrees. But in my analysis, I focused my attention on 4 of these 10 pedigrees. Why? Because of the critical numbers, genetics. My software, the software that I use, and the statistical analysis is very clear. It's when you have a very huge contrast in the number of one of the factors in the analysis compared to others, you have problems with estimations. So I avoid to have a mess in my results by concentrating my view only in those pedigrees with the largest, with a critical number, a minimum critical number of 5. This is the point. I didn't discriminate between European and North American, just discriminate by, obviously, by type of hybrid, cross type. That's it. Algunas imágenes nada más para recordarnos. Populus tricocarpa, destruido el noroeste de los Estados Unidos. Here's the distribution of tricocarpa. You already mentioned Richard and Marisque, both are described, okay, the deltoides from southeast North America. Tal como mencionaron mis 2 colegas antes, la distribución de deltoides, la zona centro-este de los Estados Unidos, cubriendo del límite con Canadá hasta el Océano Atlántico, ¿cierto?, en el Golfo de México. La otra especie que también está utilizada en esta generación de los híbridos fue Populus nigra, variedad itálica, que ya lo sabemos, Marisque nos mencionó. The other one is typical, you know, because we have Euro-American clones, D by M, so we Populus nigra. Y la última, exactamente igual como describe Marisque, es Populus maximo vixio de origen de Japón, de Asia. That was one that we used, that was included in the crosses that we tested, was Populus maximo vixio. Una cronología entonces, ¿dónde estamos? Establecimos el ensayo el año 2014, parte de nuestro programa de I+D con Café El Álamo. En 2015 nosotros empezamos a medir el crecimiento, diámetro, altura y volumen, ese era el objetivo, pero el año 2016 se detecta la presencia septoria en el ensayo, 2015 en la zona, pero ya el 2016 se presentó el ataque en el ensayo, buena oportunidad entonces para empezar a estudiar el grado de ataque de septoria. Y esto nos dio entonces el argumento para presentarnos al concurso de proyectos FIA, nos adjudicamos el proyecto. El proyecto comenzó entonces oficialmente en mayo de 2018. Just the chronology, we planted our trial to 2014, 2015 we started obviously measuring growth, diameter, height, later volume. 2016, the pathogen, we found the pathogen already present in the trees, in the stems and leaves. And since 2017, we started measuring, assessing the level of attack of the pathogen. And this gave us the good reason why we applied for the funding from the states to initiate the specific studies of the pathogen. Yeah, and this is the reason for the project. We started in 2018. El objetivo general del proyecto era generar un programa de manejo integrado para minimizar la presencia, el daño, el impacto de la especie Septoria musiva, la llamaré así por facilidad lingüística, en relación a su ataque y su presencia en el país. Entonces, el proyecto duró 2018 hasta este año. Con este seminario estamos dando por cerrado el proyecto. So this is the objective, to generate an integrated management program for controlling or minimizing the impact of the attack of Septoria in the plantings already established or to be established in Chile. Yeah. Toda la literatura habla de que la mejor forma de atacar o combatir un patógeno de esta naturaleza es a través de un programa integrado. Mauricio en múltiples oportunidades nos habló sobre ese principal enfoque. Obviously, the focus, because of the particular suggestions and orientations given by Mauricio, my colleague, is that the attack, the defensive strategy against this type of pathogen is integrated. We need to include different approaches. Por lo tanto, nosotros definimos 4 objetivos. El primer objetivo es aplicar, no diseñar ni construir nada, es aplicar. Se sabe cuándo es una estrategia de selección genética, apliquemos. Entonces, ese es el objetivo, es aplicar una estrategia de selección temprana para las variedades y encontrar las variedades que sean tolerantes, no inmunidad. Porque como menciona Marisque, la palabra es clave. No busquemos inmunidad, busquemos tolerancias, capacidad de crecer en presencia del patógeno. Esa era la idea principal. Los detalles los doy abajo de este objetivo. The objective here, the first objective is to apply, not to design, not to look for, no. The strategy is already present, we already know, so to apply a selection strategy to find trees with high level resistance, capable to growth in the presence of the pathogens. Like Mariske, I was inspired by experience in Europe. Do not look for immunity, dangerous. Look for adaptability, so to treat that grows well in the presence of the pathogen. That was the driving force for selection. Yeah. Segundo objetivo, desarrollar estrategias de control sobre las fuentes de inóculo. Estos fueron objetivos 2, 3 y 4, fueron objetivos desarrollados por Mauricio. Desarrollar estrategias, aquí sí, desarrollar. Pulir, porque el patógeno estaba por primera vez en Chile y había que buscar la forma de atacar, en forma defensiva, obviamente. Sobre fuentes de inóculo de Septoria musiva, de manera de disminuir, reducir inmediatamente la diseminación e infección de las plantaciones que ya estaban presentes en el país. Una estrategia es seleccionar las futuras, pero otra es qué hacemos con las plantaciones que están presentes. The second objective was to develop a strategy of control over the source of inocula. We already have plantings in nurseries, so the point was, okay, you select the future clones for the future plantings, or what to do within the already present commercial plantings in the region and in the country. So the idea suggested by Mauricio was obviously to develop a control, a strategy for control in those plantings to reduce the dissemination, the spreading of the pathogen. Mauricio nos presentó en un seminario en mayo pasado, el 24 de mayo, los resultados, las conclusiones de estos tres objetivos, el 2, 3 y 4. Así que lo que yo haré ahora es solamente resumir brevemente lo que Mauricio obtuvo. Posteriormente a mi término, Mauricio, si es necesario, él puede aportar algo, o recordar o corregir algo de lo que yo vaya a decir ahora. Entonces, on May the 24th this year, Mauricio gave us a talk, a seminar, where he described the results that he found and the main conclusions related to objective 2, 3, and 4, which were the objectives that he took in his hands. So right now, what I will do is just to summarize but the main results and the main conclusions. So if I made a mistake at the end of my presentation, Mauricio will correct me or will complement in case that is something is needed. Okay. Estábamos entonces en el segundo objetivo. Obviamente lo que hizo Mauricio fue estudiar, nos ilustró sobre la biología del patógeno y estudiarla como estaba presente en el país, y obviamente un tema súper importante era la forma de diseminación del patógeno. Mauricio was studying the potential sources where and how the pathogen was spreading. Yeah, and to understand the biology of the pathogen here in Chile. This was the first time that Mauricio was working with this type of pathogen attacking this type of trees. Yeah. So, nosotros también, bueno, la metodología, o no metodología, entre las principales actividades desarrolladas con este objetivo fue la captura y estudio de inóculos provenientes de cancros del fuste a través de una técnica para colectar la escorrentía a través de la lluvia, lo que sea, que estuviera fluyendo sobre el fuste, para ver si el patógeno, cómo podía eventualmente el patógeno diseminarse a través de esta vía. Okay? One of the main activities conducted by Mauricio was obviously to capture the study inocula from the canker in the stems to see how they disseminated. Yeah, it was one of the focus of the study in this objective. Second, también se estudió, liderado por Mauricio, la posibilidad de capturar y estudiar los inóculos a través del aire, en su diseminación a través del aire, con portaobjetos. Por lo tanto, esta era otra forma, o sea, no solo la escorrentía del fuste, sino que la capacidad de distribuirse en el aire. This was another activity, try to capture the study inocular dissemination via the wind, the air, not only just the potential liquid coming from the cankers and the stems, but also the way that was disseminated in the air. There was another approach. También realizó pruebas de patogenicidad con aislados de Septoria Musiva recolectados en el campo en estos ensayos y en plantaciones jóvenes, para poder evaluar, bueno, el grado de patogenicidad, cuán virulento era el patógeno. Obviously, taking into consideration what was Mauricio collecting in the field, in the trials and in young plantings, he also conducted some pathogenicity trials in nursery conditions on campus to see how the pathogen, try to classify, obviously, classify the level of pathogenicity of the pathogen. También acá otra de las grandes actividades que se desarrolló, el control de inóculos en hojarasca, es decir, limpieza, y si recuerdo bien también la posibilidad de urea, algún otro tipo de producto, cosa que pudiera eliminar o acelerar, por ejemplo, acelerar la descomposición de la hojarasca, para poder limitar la permanencia del patógeno en terreno y su diseminación. This is another activity, is to start to observing the cultivation practice, the management practice, to clean up, to to avoid the dissemination of the pathogen under planting conditions, and using urea, for example, if I remember, and to try to see if we could reduce, to accelerate the composition of the degradation of the leaf litter. So we control the dissemination of the pathogen by some management practice in plantings. El objetivo 3 fue evaluar la eficacia de biocontroladores sobre el crecimiento in vitro del patógeno. The second objective, activities connected, the second objective was to assess the efficiency of biocontrol in vitro in Septoria Musiva. Aquí no obtuvimos grandes resultados, se obtuvieron resultados, pero no se obtuvo la meta que se perseguía de poder inhibir la incidencia del patógeno, de tratar de controlarlo. El patógeno es duro de matar, cuesta hacerlo. Aquí están mencionadas esta imagen sacada de la presentación de Mauricio, se mencionan los biocontroladores principales. Si recuerdo mis conocimientos, es una bacteria, bacilos y 2 hongos, los que fueron probados. In the case of the biocontrol testing, we didn't get all what the objective was accomplished, but we wanted to get a better result. That was more or less the idea. It's very hard to kill this pathogen, and very, very strong. Mauricio tested 2 fungi, according to what I see here, two fungi, 1 species and 1 type of bacteria, and the results were not, well, okay, but not promising, in the sense that it's very hard to control via biocontrollers. This is the main conclusion that we could afford mention right now. Ahora, el objetivo específico número 4, desarrollar un programa de control químico para disminuir la diseminación del patógeno en plantaciones y viveros sanos. The last specific objective, to develop a chemical control program to diminish the spreading of the pathogen in plantings and commercial nurseries. Se probaron distintos tipos de aplicaciones químicas en vivero y plantaciones para generar un programa de aplicación. Different type of chemicals were tested in nursery conditions in the same company, and also in some young plantings, and different products and different dosages, to try to see which one was the best to give us the best results, and to minimize the presence in nurseries and plantings. Todos estos resultados y las conclusiones principales de Mauricio quedaron registradas en un documento, el cual hemos llamado Manual Técnico, Método de Control Integrado. Es decir, todos estos objetivos 2, 3 y 4, particularmente resultados del segundo y cuarto objetivo, porque son prácticas de manejo, están descritas en un documento que es un principal resultado o producto de esos 3 objetivos a través de este proyecto. The main result and the conclusions and the practice that are suggested by Mauricio to be implemented for the control of the pathogens in plantings and nursery were summarized in a document, this technical document, technical manual describing the different methods to control the pathogen. Yo ahora voy a describir en varias imágenes lo que no fue descrito antes, es decir, los principales resultados obtenidos y conclusiones de la selección genética. Now I'm going to describe the main results, conclusions, what we observed, what I detected by trying to understand the genetics of the attack of the pathogen and the effect and the growth. This is what I will show right now. Y obviamente mostrar el catálogo, el principal resultado, un catálogo de variedades que en presencia de patógenos pueden crecer, desarrollarse, and also to show the catalog, the set of clones that I selected, varieties that in the presence of the pathogen are able to grow. Nosotros empezamos en nuestra evaluación de todo lo que estuviera disponible en nuestras manos, que era parte de nuestro programa de trabajo con la empresa. Eso incluía algunas plantaciones demostrativas y en particular el ensayo que yo mencioné, de 124 variedades de clones. We started studying the presence of the pathogen and the effect on the growth in any possible available material that we had from our program, research program with the company, including the clonal trial that I already mentioned, established in 2014 with 124 varieties, but at the same time we were having some demonstration stands with some of our clones, and so we will also observe the attack and in those conditions. Utilizamos, obviamente había que incluir el año 2017, como mencioné, la cronología al año 3 de plantación. Nosotros empezamos a evaluar la presencia de patógeno y básicamente, según la literatura, una escala de 3 grados. La escala, el grado 0 obviamente es no ataque, no hay presencia de ataque. El grado 1, presencia de exudaciones, bien específico, ¿eh? Presencia de exudaciones y 3 cancros de cierto diámetro, y el grado 2, presencia de más de 3 cancros, y bla, 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 bla. Entonces, esa fue la primera forma de clasificar. The first, at age 3, we use a kind of the standard classification of the attack, and the level 0, no attack, and then we increase. The increase of the level of attack is graded 1 and 2. Al año siguiente, ¿qué es lo que hice yo? Dado que había mucha heterogeneidad con los cancros atacando 1, 2, 3, lo que sea, a distinta altura, entonces dividimos la escala. Ya no fue ya un 0, 1 y 2, sino que ahora fue 0, 1, 2 y 3. El nivel 2 lo separé en 2, para separar un poco esto de las distancias o el número de cancros. At age 4, I split level 2 in 2 and 3. Just to be a little bit more specific, because there was different level of attack. The attack was very complex, and we were learning as we were observing, evaluating. We learned the way that the pathogen was expressed in the stem. Nos concentramos en el fuste, eso sí. We focus our attention on the stem of the tree more than the leaves, because the economic value of the company was located in the stem. Debido al valor económico que tenía el árbol, lo tiene, para la compañía lo tiene en el fuste, no en las hojas. Entonces al año 6,5 y el año 5 comenzamos a evaluar según una nueva escala. Ya voy a decir por qué 6,5 de evaluación, pero partamos. El nivel 0, al igual que en el caso anterior, no ataque presente. Es decir, si el fuste estaba limpio, aunque pudiese haber tenido ataque el año anterior o 2 años antes, si este año tenía el fuste limpio y uno podía detectar ningún tipo de exudación o cualquier tipo de presencia de patógeno, entonces era evaluado con nota 0. It doesn't matter if the attack with the previous year or 2 years before, there was some attack in this individual. If the stem was clean, no signs of exudations, obviously, of any sort of attack, the tree was graded 0, no attack. Pero el año 2020 nos ataca el COVID y eso me cambia el esquema de hacer evaluaciones, por eso ya no hay evaluación al año 6, sino que tuve que posponer la evaluación y caemos al año 6,5, no es un gran drama. Due to the presence of COVID in 2020, I had to postpone the evaluation of the trials. So for that reason, I talk about evaluation at age 5 and at age 6.5, was not at 6, 6.5. Ahora, esta es parte para las evaluaciones del año 5 y 6,5. Yo utilicé una escala que yo mismo desarrollé. Partamos por, esta escala tiene un número 1 y una letra, pero tiene un número y una letra. El número 1 significa que estamos analizando las exudaciones. Es exudación. ¿Qué es lo que significó la letra? La letra A es que tenía una sola exudación. Un individuo 1A, es decir, le encontré exudación, pero una sola. I coined this type of classification, number 1 for exudations. number one for exudation. The letter A means I detected only one exudation, nothing else. In the full stem, visible stem, only one exudation. Uno ve exudaciones, pero dos o más. 1B, we have exudations, but 2 or more, but only exudations. El nivel 2 es cuando tenemos cancros. 2A cuando tenemos un solo cancro. No importa si hay 3, 4, 5 exudaciones, pero si tenía un cancro, uno solo, 2A. Level 2 means cancers. Letter A is one cancer. Doesn't matter if there were several exudations, but if there is one cancer, so that indeed was specified 2A. Y obviamente el 2B es cuando es caos. 2B es, tengo cancros, pero tengo más de 2 o más. 2B is that it was cancers, but 2 or more cancers present in the stems. This is level 2B. Esta clasificación, esta forma de evaluar la presencia septoria en el fuste fue otro producto. Esto se elaboró un breve documento describiendo esta clasificación. This is another product that we obtained with the project. A kind of brochure, a brief document describing the classification, the way of detecting this, the pathogen in the stems. Yeah. ¿Qué caracteres estudié? Obviamente esto es de valor comercial, es el diámetro, la altura, por lo tanto el volumen, y le agregamos entonces la intensidad del ataque en el fuste. These were the characters of obviously the characters with the economic value for the company is the diameter, total height, volume, and I added the level of attack of the pathogen. Esta tablita de doble entradas me describe a mí the field experimental design, es el diseño de experimento. Es decir, yo tenía cuatro cruzamientos, dentro de cada cruzamiento tenía 5 o más variedades plantados en 20 bloques, pero yo ahora le agregué, como medí el grado de ataque, se lo agregué. ¿Ya? Y entonces salté, en vez de estar estudiando lo típico en los estudios de patogenicidad, yo voy a decir la intensidad del ataque, tantas esporas o lo que sea, por ejemplo, aquí es distinto. Cada individuo fue clasificado según el grado de ataque. Entonces lo que yo quería tratar de evaluar era el efecto del ataque en el crecimiento. This table describes the experimental design. We have 20 blocks and we have 4 crosses, and within each cross we have a different number of clones, 5 to 71. And because I measure because I assessed the level of attack, I also included in the table, in the classification, as a classification factor in statistical point of view. So what is the point here? That instead of measuring as the characteristic to be the number of inocula, whatever from the pathogen, no, I study the growth, diameter, volume, whatever, in the presence of the pathogen, to see if I could isolate the effect of the pathogen for selection purposes. This is what I did. Entonces, esto es, no los voy a marear con esta cuestión, solamente para mostrar que esto que está acá arriba es el típico representación matemática en el cual se basa el análisis de selección. Tengo la característica dependiente, mi variable, mi fenotipo y los efectos que están actuando sobre. Eso es lo que trata de decir, los efectos que están actuando sobre el carácter, el cruzamiento, el bloque, el espacio microambiental, etc. Sin embargo, yo le agregué acá el efecto del patógeno. Como lo clasifiqué, el efecto de ataque, lo incluí. Obviamente esto hizo todo mucho más complejo. Sí, pero es posible analizar. The original linear model that describes the phenotype, the typical, of course, we have crosses, clones, microenvironmental effect, etc., etc. But because I was able to measure the presence of the pathogen, I included in the model. it produces more complication. Yeah, it's more complicated, but still you are able to do the analysis. The model becomes more complex to analyze, but it's possible. Ahora apliqué el principal criterio de selección, es lo que se conoce habitualmente con esta sigla, incluso en español lo hablamos así, los VLUP. ¿Ya? Los VLUP, ¿qué significa? Es una regresión a partir de los datos para predecir qué cosa esto que está en el círculo. Eso es el que tiene valor genético. Yo quiero predecir el efecto del clon. ¿Y qué es lo que hice? En presencia del patógeno. Eso fue lo que yo quise atacar. Quise predecir el valor numérico de este clon en presencia del patógeno. I used these techniques that is the standard for genetic selection, trees and plants in general, animals, the best linear bias prediction, a kind of regression, and I predicted the clonal value in the presence of the attack of the pathogen. That was the idea. Utilicé los típicos análisis que corresponden, que hay que realizar en esto, para los 4 años, los distintos 4 años de evaluación. Los principales resultados de esto fueron publicados ahora. Yo estudié muchas más características, pero preparé un artículo que fue publicado ahora en junio, donde yo doy a conocer la metodología, la innovación de la clasificación del grado de ataque, etcétera, y el artículo fue publicado. Este es un producto de comunicación científica del proyecto. I did analysis, I started testing the use of my classification for the pathogenic attack. So by doing the analysis of one of the characteristics that I selected, I prepared this paper. This is one of the products of the project, the scientific communication, just published now in June, last June. We are fine. Y este es el tipo de tabla. Obviamente no voy a entrar a especificar el detalle, pero es el tipo de tabla. Y ustedes ven aquí, se alcanzará al año 4, al año 5. Yo hice mi selección con distintos números de individuos seleccionados y utilizando como principal criterio esto que se denomina VLUP, esto que está puesto acá en esta representación, con letras muy extrañas, con negritas, son matrices, vectores. Y esto es, aquí están los valores fenotípicos, crecimiento, y esto que está restado acá al lado está metido a los efectos del patógeno, es decir, castigado, corregido por la presencia del patógeno, y eso me da entonces un valor genético de cada variable. Eso en términos lo más simple posible de describir en qué consiste esta cuestión. And we can observe here that this is the representation, algebraic representation of matrices and vectors of what is this difficult to see, but this is a regression analysis. And here what we can see is the Y vector is the phenotypic observations. And here minus X by this other type of beta, whatever. This is the effect. Here you can see the effect of the pathogens. Because in other words, I am correcting the phenotype based on the presence of the pathogen. This is the idea that I had in mind when I included the pathogenic effect in my analysis. Voy a mostrar algunos valores, lo esencial de lo que yo observé, no en todas las características, pero en volumen, que es lo más relevante. Quiero describir brevemente este gráfico para que ustedes vean como resultado y conclusión también de lo que se observa del ataque del patógeno, que creo que esto es relevante. En el eje horizontal vemos el nivel de ataque. ¿Qué significa el nivel de ataque? Cada punto que observan ustedes en este gráfico es una variedad. Los números horizontales van de 3, 4, 5, 6, 19. ¿Qué significan estos números? Es por ejemplo, esto que estoy indicando con el mouse acá, esta variedad tuvo, cuando lo evalué al año 3, tuvo 18 árboles de la variedad sin ataque. 18 de 20. Este otro individuo que está acá tuvo solo 5 árboles de 20 sin ataque, el resto atacados. Entonces esto significa horizontal hacia la derecha, menos ataque, y hacia la izquierda, más ataque. En el eje vertical el crecimiento, ¿cierto? Más crecimiento, menos crecimiento. La primera línea roja, algo se alcanza a ver, yo creo, es el primer cuartil. De ahí a la izquierda, 25% de los valores. Esta otra línea dice acá 13, ese es el tercer cuartil. De acá hacia el lado derecho, 25% de los datos. Entre medio, 50%. De igual manera para volumen en el eje vertical. La línea punteada es probable que no se alcance a ver bien. Ese es el valor promedio. I'm just describing briefly the plot in the horizontal X. You will see 6, 5, 10, 11, 12, 19. What is the meaning for that? For example, this dot here, this circle, is one variety. Here, this variety had 18 out of 20 with no attack. No attack, clean. 2 were attacked. Now to the left, this other one here to the left is only 5 trees, no attack. 15 with attack, which means more attack, less attack. Vertical X, volume, less growth, less volume, more volume. The first line here is the first quartile. The second red line continues the third quartile. So we have to the right from here, we have 25% of the data. From the first quartile to the left, 25% of the data. Here in the middle, 50%. Y por lo tanto, nosotros vemos, aquí está el cuartil que más me interesaba. Es mayor crecimiento, menor susceptibilidad al ataque. ¿Qué observamos rápidamente en los teclos D y los teclos D? A medida que aumenta el crecimiento, mayor ataque. A medida que disminuye el crecimiento, menor ataque. El patógeno empezó a atacar, vimos evidencia que atacaba a los que crecían más. En otras variedades, Decrus N no era muy claro. Los puntos en rojo acá son los clones comerciales europeos. Los puntos en azul que están indicados con las flechas de acá son clones comerciales, variedades comerciales norteamericanas. Buenos crecedores, pero más susceptibles a ataque. So what we can see in this plot is the clear tendency that as the variety was growing and growing and growing, was more and more susceptible to the attack of the pathogen. The less that the tree grew, less susceptible. More trees will not attack. That is a clear tendency in T by D. Not so clear in D by N, Euro-American clones. These red arrows here are North American commercial clones. These other 3 arrows are European commercial clones. I-488, I-214, the typical clones, commercial clones from Europe. Saltamos al año 4, lo mismo, la misma tendencia. At age 4, exactly the same. The larger the growth, the stronger the attack by the pathogen. A medida que crece más el árbol, aumenta el nivel de ataque. Mientras crece menos, menos presencia del patógeno en el fruto. El año 2019 se presentó la mayor intensidad de ataque y ustedes observan una relación muy fuerte entre uno y otro. Claramente se observa mayor crecimiento, mayor nivel de ataque. Here in 2019, the evidence was this phenomena, the correlation, negative correlation, was extremely clear to see. The larger the growth, the speed, the growth speed, the stronger the attack. Less growth, less attack. Bueno, y esto es el año 6,5 exactamente, 6.5, age 6.5, exactly the same trend. Ahora, eso fue solamente con los individuos que tenían nivel 0, pero ahora yo los desparramé. Esto es muy breve de mencionar para que ustedes vean claramente que a medida que aumentaba el ataque, el crecimiento aumentaba el ataque. This plot right now is just the box plot for all the measurements at age 3, just for you to see what I was just describing. The faster the growth, the upper the level, the higher level of attack. Mientras mayor era el crecimiento, mayor era el nivel de ataque. Todos estos individuos que están indicados aquí en el círculo son de los distintos cruzamientos, tienen mayor crecimiento comparado con los individuos que tenían nivel de ataque 0. You can observe here the higher the level of attack, correspond to the trees with the largest volume. Esto fue el año 3, el año 4, the same and age 4, exactly the same. Here we have level 0, no attack, mainly for trees that are growing less. Well, the values are spread, it's true. Yeah. But we also observe that the faster the growth, more attack. El año 5, 2019, exact, age 5 is very clear, right? The level of attack 0, slower growth, level of attack 2B is the trees that the faster grower, the more accumulated growth are more susceptible to attack of the pathogen. A la edad 6,5 exactamente igual, 6.5 exactly the same. Yeah, just a conclusion. So, ahora que lo que para concluir entonces este resultado que creo que es relevante es estas edad 3, 4, 5, 6,5, que es el factor 0.5. Estos son los promedios para los distintos grupos 0, 1, 2, 0, 1, 2, 3. Y acá viene 0, 1A, 2A, 1B, 2B. Así es la manera como está clasificado aquí, está indicado. Y obviamente ustedes ven los que tienen menos crecimiento son aquellos que están en categoría 0. Menos crecimiento, 0. Y a medida que aumentaba el ataque, 0, 1, 2, 3 o 0, 1A, 2A, 2, 1B, 2B, correspondía a individuos que crecían más. We see ages 3, 4, and 5, and 6.5. Made a tiny mistake here. The first column, level 0, no attack. As we increase the attack, using my classification, the one that they rate, it was very good to discriminate the growth. I didn't know that when I did it the first time, but you can see that as the stronger the attack, the faster the growth, the larger the amount of body mass of the tree. Yeah. Ahora, esto viene a la pregunta, ¿hay diferencias significativas entre estos? And the question is there statistical significant differences? Yeah. Aquí está un resumen, diámetro, altura y volumen, todos los test estadísticos, todos, el error tipo 1, la típica cuestión, todos me dicen extremadamente estadísticamente significativas las diferencias. Is diameter, height, and volume, all the statistical tests, and the p-value here at the extreme right. So all of them highly significant, meaning that I was able to separate the kind of effect of the pathogen from the growth. Yeah, and there were very strong statistical differences between groups. Entonces utilicé esa información. Me sentí más tranquilo y utilicé esa para hacer la selección. Ustedes ven ahora en el eje vertical ya no es el volumen, sino que es el valor del value P, así denominado. Entonces, valor 0 está indicado acá, porque como son desvíos, está corregido, entonces algunos valores son positivos y otros negativos, y eso inmediatamente usando versus el eje X, que ya lo describí. Entonces, yo ataqué entonces este cuarto superior derecho, donde están aquellos individuos con menos susceptibilidad, más resistencia a patógeno y mayor valor. Okay? I use the BLUP to classify the individuals. Here we see it in the YX, the value 0, Y is 0, because it's an adjustment. So you have trees with a value positive or negative. And the horizontal X is a typical that I already described. So I focus my attention on this section, the section of the trees with the largest value and with the small level of attack or the highest resistant value. I will put it that way. Entonces fui seleccionando para cada edad exactamente lo mismo, aquellos que estaban ubicados en este sector superior derecho. ¿Ya? For each year of analysis, I start to select the trees which were located in that quarter, in that section Yeah? Larger value, BLUP, clonal value, and the larger level of resistance, less susceptibility to the pathogen. Exactamente igual para cada uno de los años. Este es el caso de volumen. No, obviamente que acá todos los clones comerciales norteamericanos tenían buen valor, pero muy susceptibles. Just this plot is showing me here all the commercial North American commercial clones, a good, very high value, genetic value, but extremely susceptible to the attack of the pathogen, all of them. And I was able to select some that were having a reasonable good level value, but at the same time, much less susceptible to the attack of the pathogen. Yeah. Y al final, entonces, construí estas tablas en base a los valores para el volumen, pero a su vez también detecté aquellos individuos 37 que en todos los años, a distintas edades, porque estudié 3, 4, 5, 6, 6,5, aquellos que tenían al menos un 80% del material genético, árboles, sin nivel de ataque, entre 80% y 100%. I also selected 37 individuals that were able to grow from 1 year to the next with absolutely no attack, or at least 80% and more of trees with no attack at all. Entonces, principal resultado, el catálogo. Ya mencioné cómo se estructura el catálogo, las tablas están disponibles. 8 variedades seleccionadas a la edad 3, 10 variedades seleccionadas a la edad 4, 5 variedades seleccionadas a la edad 5, 7 variedades seleccionadas a la edad 6 y 37 variedades seleccionadas por inmunidad, por un porcentaje de inmunidad. No al 100%, pero inmunidad, es decir, que no presentaban ataque al momento de ser evaluados. This is the main result of what I got. 8 hybrid selected at age 3, 10 selected at age 4, 5 selected at age 5, 7 selected at age 6, and 37 selected by their resistance to the pathogen. Para terminar, las últimas 2 imágenes, nosotros importamos también material genético desde Canadá, de Quebec, del Ministerio de Bosques, Fauna y Parques, 18 variedades, las cuales están ahora, ahí están los pedigríes, 18 variedades, distintos estacas de cada una de las variedades, y esas variedades ahora están en un populetum dentro de la empresa forestal Café del Álamo. Yeah. Just the last 2 images showing what we imported 18 clones from the Ministry of Forest and Animals and Park from Quebec, Canada, and this material today is in a nursery in the field, in the company, and they will be established in clinical trials to continue the testing and selection. This material was selected for the resistance to Septoria in Canada and until the year 2030. This is the program. Este material va a ser establecido ahora. Hay un retraso debido a la pandemia. Y este material tiene que ser establecido este año para ser evaluado. Hay un desfase de 1 año, hasta el año 2030, en que se deberían tener ya resultados por grado de resistencia al ataque del patógeno. Para terminar, quiero agradecer entonces al gerente de la empresa, Jaime Venegas, a Luis Retamal, jefe forestal. I want to thanks to my colleagues from the company, the forest manager and the head of the forest department of the company that were extremely supportive during all these years that we collaborate with them. Obviamente agradecer esta colaboración que hemos tenido con Richard y Nicolás. Thank you, Richard. Thanks to Nicholas for our cooperation with both of you. ¿Ya? Y finalmente quiero agradecer a mis colegas y amigos que me han acompañado durante todos estos años en esta investigación y otras que hicimos anteriormente. I just want to thank my friends, my colleagues, my collaborators from all these years that we conducted not only this research, more research. ¿Ya? Fernando, Jorge, Mauricio, Marco, Javier, Cristian y la señora Carolina Vila. Y por último, ahora termino. Muchas gracias a Maurice Strait, el ejecutivo de nuestro proyecto. Just to finish finally is the person or the agent who was supervising our project in this foundation that support our research. Yeah, thank you. Ahora he terminado.
Excelente, profesor. Muchas gracias. Fue una jornada que se hizo muy breve porque todas las presentaciones fueron muy bien expuestas y muy resumidas y se le agradece a cada uno de los expositores por ese esfuerzo de condensar tanta información en un lenguaje fácil de entender. Con esto les damos la bienvenida, o sea, la despedida. Muchas gracias a todos por ser parte de este proyecto y será hasta una próxima oportunidad. Muy buenas tardes a todos.
Ya, gracias.